So Laboratory 1 is relatively straightforward, and you can get all the information you need from the introductory lecture and also from the lab book. So I'm going to start here with some info from lab number 2, which has to do with dissection. So this is a dissection of a fetal pig, and I just wanted to start by pointing out some of the key organs. So in class, when we would dissect this is what it would look like. And so if you start here, this is the heart. These are the lungs on either side of the heart. Sometimes on the heart you can see the thymus. The thymus is a gland. It's part of the lymphatic system. That may be a portion of it right here. And coming up here in the neck area, you can see what looks like is the trachea. So the trachea is the airway, or sometimes it's called the windpipe, allows air to get down to the lungs. And it has these little ridges on it. So it has these little rings of cartilage on it, so it always has a bumpy appearance. Behind the trachea you would find the esophagus, which you can't really see that well here. This is the liver, so here and here. So the liver is really big. And then beneath that you see the stomach. So here's the stomach right here, right here. It looks like a balloon that's been deflated. So that's what the stomach looks like. Kind of wrapping around this side, this is the spleen. So here, spleen, and the spleen is also shown here. And then as we make our way down, you see lots of intestines. And I'm going to move to a different picture that you can see a little bit, some different views here. But I wanted to point out, you can see one of the kidneys right here, and you can also see it right here. So the kidneys, you can have two kidneys, one here and one on the other side. They're just very deep. They're in the lower back area. So you can't really see them that well with all of the intestines in the way. So here, another good picture of the stomach, and these are all the intestines. So stomach, spleen, intestines. And then a little bit closer look, here's your two kidneys that I was talking about. And so those are the big structures that you need to be familiar with for exam purposes in this dissection. So you might see pictures like this on your first lab exam with labeling, and you should know what the structures that we just went through. So only the structures that I'm covering right now. So to do this again: heart, lungs, trachea, spleen, stomach, liver, intestines, kidney. And here we see the kidneys again, stomach, spleen, intestines. There's the liver too. So the next lab number 3, has to do with the microscope. So this little bit of information I'm going to give you along with the microscope lecture will be really helpful at answering some of the lab questions about in the microscope lab, and also they're fair game for the first lab exam. So this is what the microscopes that we use look like. So in the previous lecture I went through this, but I'll do it really quickly. You have your ocular lenses, your objective lenses that each have their own magnification. You have the stage with the little clip that holds the microscope slide in place. The condenser right above that, in the iris diaphragm aperture that allows you to change resolution and contrast. Here's your fine focus and coarse focus adjustment knobs. Here's your X and Y axis movement knobs that'll move the stage either right to left or forward backward. Here's the light source. So what's missing in this picture is the rheostat looks different than what ours normally looks like, but this is the rheostat right here. It controls the light intensity. And then there should also be an on-off switch somewhere in here. So running through the magnifications again really quick. The ocular lens always have a magnification of 10. Scanning lens, which is the shortest objective lens, is going to have a magnification of 4. Low power is going to have a magnification of 10. High power, magnification of 40. Oil immersion, magnification of 100. So to calculate the total magnification, which you'll be expected to do, you multiply the ocular times the objective. So if you want total magnification through the scanning lens, it would be 10 times 4. So 10 comes from the ocular, and then the 4 comes from the objective. So together that gives us a total magnification of 40. I'll just jump down to high power. So we still have 10. It's always going to be 10 for the ocular and 40 for the objective. So that gives us a total magnification of 400. So another key thing that you'll learn as you go through the lab is as you move to higher magnifications, you need more and more light. So as you would move to a higher magnification, you'll have to increase the light intensity a little bit to see clearly. So one of the lab activities is telling you to look at the letter E through the microscope. And so there's a microscope slide with the letter E on it. So when you actually look through the microscope, here's your field of view right here. And so the diameter, we'll talk about the diameter of the field, that's from here to here. So this is with the scanning lens, total magnification of 40. Notice what the E looks like. When you put it on the microscope slide, it's actually right side up. But when you look at the microscope, everything is reversed. So everything looks upside down. That's just the way it is. So you should understand that if you look through a microscope and you have it on the slide one way, through the microscope lens, it's actually going to look the opposite. And so as you zoom in to move to the low power lens, which would be 100 total magnification, you can see it gets bigger, as you would expect. Then as you get to like 400, using the high power lens, you can't even tell it's an E anymore because you're just kind of zoomed in on one of these areas. So that's one of the lab activities that you'll see in your lab book that I'm trying to just give you an idea of what you would see. So you should understand that concept that what you see is upside down. Then also, you can also tell as you go up in magnification, you're going to need more and more light because everything seems to be a little bit darker. The other good thing to take from the microscope lab is the diameter of the field. So like I said earlier, when you look through the microscope, you'll see a circle like this. This is your field of view. We generally want to know how big that field of view is. So we calculate the diameter from here to here. And so one way we do that in the lab is we have slides with graph paper. Each little box is a millimeter. So with the scanning lens in place, with a total magnification of 40, it's about 1, 2, 3, 4, 5 millimeters across. So the diameter of the field with the scanning lens in place is going to be 5 millimeters. If we zoom in with the low power lens, and now we have a total magnification of 100, it's going to look like this. So now you can see our diameter has completely dropped. So now we have a diameter of the field of 2. As we go to 400, it's going to be even less, right? Not even 1 millimeter. So key point, as magnification goes up, diameter of the field decreases. And sometimes it gets hard to calculate the diameter of the field because it's not perfect. So there's an equation that is discussed in your lab that you can use to calculate this. So it's basically solving for an unknown. So if you know the diameter of the field of one lens, and I'll say it's A versus B, and it could be any two lenses, any two objective lenses. So diameter of the field of lens A times total magnification of lens A equals diameter of the field of lens B times the total magnification of lens B. So, and if we know 3 of these things, we can calculate the 4th. And so I'll demonstrate that on the next slide. So if we want to know the diameter of the field of a given lens that we don't know, we can use what we do know to figure it out. So in this example, if we set the scanning lens as lens A, and this is just arbitrary, and the low power is lens B, what's the total magnification of the low power lens? So 5 would be the diameter of the field of the scanning lens with the scanning lens in place. We've established that by looking at the graph paper. Total magnification with the scanning lens in place is 40. We've established that. We want to know the diameter of the field of the low power lens, and we know the total magnification of the low power lens, which is 100. So then we just use some basic algebra and solve for x. So 5 times 40 is 200, 100x, and then divide both sides by 100 and solve for x, so we get x equals 2. So we calculated this and we got 2 being the diameter of the field of the low power lens, and just to show you that was accurate. because we can actually go back and look at it with graph paper. So we can do this either visually or by calculating it. So sometimes with the high power lens, like we saw, it's kind of hard to tell what it is just by looking visually. So I'll do another example right here. So to calculate the diameter of that high power lens, we can set the scanning lens as lens A again. We could use low power if we wanted, it doesn't matter. And then the lens that we want to find, we put here. So high power lens has a total magnification of 40. We want to know the diameter. So you get 200 here again, 400x, divide both sides by 400 to solve for x, and we get that x equals 0.5. So that means the diameter of the field with this lens in place, with the high power lens in place, is 0.5 millimeters. So this is important because once you know the diameter of the field, you can estimate the size of any object you're looking at. So in this example, let's say this is the field of view, and we didn't calculate this, or let's say, well, it's saying 2 millimeters, so we're assuming this is a high power lens, but I'm going to trust that this is 2 millimeters from here to here. And let's say we're looking at a cell that's this big. So what we can do is just kind of estimate that this cell looks like it's taking up about a fourth. That's my guess, about a fourth of this diameter. In other words, if we had 4 of these cells, it would take about 4 of these cells to make it across. It may not be perfect, but it's close. So if this cell is about a fourth of the diameter, then we can estimate that the cell is about 0.5. So all I did was divide the diameter by 4 because we assumed this was about a fourth of the diameter, and I got 0.5. So we can estimate that this object is about 0.5 millimeters. So key point, the importance of knowing the diameter of the field is that you can use that to estimate how big an object is. All right, and the last thing I wanted to bring up that's related to the microscope lab is the concept of working distance. And all working distance is the distance between the objective lens and whatever you're looking at, a microscope slide or object. So you can tell from this picture that the working distance, which is this, gets smaller as the objective lenses go up in magnification. This is one of the reasons that you don't want to use the coarse adjustment knob with any other lens other than the scanning, because you could easily break the microscope slide by ramming it into the objective lens.